Second messengers.
Second messengers are molecules that are found in the cytoplasm of a cell, which trigger biochemical communication between other cells. These communications can be responsible for long-term changes in the nervous system. The second messengers tell the chemical message of a neurotransmitter to the cell's internal biochemical machinery. The effects may last from a few milliseconds to many minutes.
There are three types of second messenger systems. The cyclic adenosine monophosphate system, also known as CAMP system, phospholinositol system, and the acryazotic acid system. The most common messenger system is the CAMP system. This second messenger system involves ATP, the chemical source of energy in a cell. When noradrenaline binds to its receptors on the neuron, the receptor will bind a G protein to the inside of the membrane. A G protein is a protein that transmits information outside of the cell and causes changes within the cell. The G protein then causes the enzyme adenyl cyclase to convert ATP to CAMP.
The newly created CAMP will then bind to and activate a target protein, for example, alpha kinase, and will then add phosphates to the proteins in the cell. The effects from this pathway will depend on the identity of the cell and which proteins are phosphorylated. Another pathway is the IP3 pathway. The primary function of the IP3 is the release of calcium from the endoplasmic reticulum from the IP3 receptor. The release of calcium from the ER is an important signaling event in all cells, and it must be regulated within the context of specific cell needs. CAMP has several different influences on the cell, such as the function of ion channels or the expression of genes in the nucleus. Other second messengers are involved in the manufacture and release of neurotransmitters, and some deal with intracellular movements and carbohydrate metabolism in the brain. Direct effects of second messengers on the genetic material of cells may lead to long-term alterations in cellular functioning and ultimately in behavior.
These long-term alterations that Matthew was talking about includes disorders such as manic depressive disorder, MDD, and Tourette's. The phosphodilynositol and calcium ion second messengers are of particular interest because of evidence of being extremely active. Of these signaling mechanisms in both mania and bipolar depression, of their normalization by lithium and other mood-stabilizing treatments.
Within the cell, there is the Ca2+ signal that has a biphasic action. A tiny abnormality could be the cause of many clinical situations, and a single action on the messenger could be the explanation to the biphasic treatments for the bipolar disorder. Second messengers have also been suspected to be the cause of Tourette's syndrome. Research on this study has not been clarified or elaborated on, but several cases have stated that an abnormality in the system has been the cause to the disorder. The anatomy of second messengers cannot be displayed because they are intracellular. On the other hand, second messengers send out signaling messages. Cells must be ready to respond to essential signals in their environment. These are often chemicals in the extracellular fluid. There are three types of signaling messages: the endocrine, paracrine, and autocrine. A cell signals itself through a chemical that it synthesizes and then responds to. Autocrine signaling can occur within the cytoplasm of the cell, or by a secreted chemical interacting with receptors on the surface of the same cell. The paracrine system sends chemical signals that diffuse into the area and interact with receptors on nearby cells. It is like the release of neurotransmitters at synapses in the nervous system. In the endocrine system, the chemicals are secreted into the blood and carried by blood and tissue fluids to the cells they act upon. These signal messages can trigger an immediate change in the metabolism of the cell, in the electrical charge across the plasma membrane, or in the gene expression.
In conclusion, second messengers are a very important part of the human body because of their ability to communicate with receptors within the cell to relay certain hormones. Second messengers are a key aspect that makes us who we are and holds our bodies together.
And without them, we would most likely split in two, just like this handsome young cell is doing in the image displayed.
Ha 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 ha! Good one, Josh. Hope you enjoyed our presentation.
And without them, we would most likely be split in two, just like the handsome cell is doing in the image display.